close the door behind you, please. Okay, good morning all of you, as there are not many of you, but still. I am Dr. Deeba from Department of Medical Biochemistry, and I will be teaching you the third portion under the course of General Biology. Now, when you started the course, you went through cell biology. Majorly that cell biology was structural biology, where you learned about the structure of different components of the cell, maybe a little bit about transport and all. And then you had molecular biology, talking largely about DNA, RNA and proteins. Now, under my portion, again it is cell biology, but we are going to build a level up on what Dr. Sama has taught you. So, largely Dr. Sama has discussed the structural components. We will look into excuse me, the functional component of the cell biology. So, we are together for roughly 3 weeks with the midterm in the middle somewhere, I think from next week onwards. So, this is the list of the topics that we will discuss together, starting with protein trafficking or protein transport, which deals with the proteins that are made in our cell. How do they go to their different destinations? Who decides which protein will go where? Then it will be followed by protein degradation. Degradation means breakdown. So many proteins are made in the cell. Are all of them kept until the cell is alive? No, they are. When they are not in use, they are degraded. So, what is the system, how it is done? We will talk about that. Then cell cycle. Our body is made up of cells, and those cells undergo different phases to maintain their structural and functional component. And under cell cycle, we will learn what are those phases. And since the health of the cell is important for the health of the human being, we will look at how closely and strictly the cell cycle is regulated. After that, we will come to signal transduction. Now, all of you structurally know that on cell membranes there are receptors, and when one cell needs to communicate with the other cell, a molecule comes and binds to that receptor, correct? However, under signal transduction, we will look at how that signal that is bound to the receptor is processed inside the cell, how it is converted into an action in the cell, okay? Followed by cell death or apoptosis. So, there are certain conditions in which cell chooses to die rather than live, and that is called as apoptosis. What is the process, how it is done is covered under this topic. And finally, the last topic is aging or senescence. Now, as humans, we are born, we grow up, and then we slowly start aging, correct? And you can see those changes in everybody. But when those changes happen, years before that, the aging starts at the cellular level, and that is called as senescence. So, we will talk about that in the last resource that I will have with you. Okay. Now, before I start on with today's resource, I want to give you a few guidelines which about how my sessions goes on. So, and few of the questions that I have heard over the years, student asking different instructors. So, the most frequently asked question is, Doctor, are we responsible only for the slides, or it is going to be book and slides? So, for this particular doctor, that is for me, whatever I am teaching, you are only responsible for the slides, for the exam, okay? However, as a specialist myself, and you are going to be future doctors and specialists, it is always better to go back to the book and see the same topic, okay? But as far as exams are concerned, slides are enough, Also, if you will look at my slides, the light is too much, I do not know how will you see all of it. You will see a diagram and you will see text. Me as an instructor, I hardly read the text, okay. This text is for you to go back and read. I mainly focus on the diagram, and trust me, if you are there and going with me through the diagram, it will be more than enough, okay. Another thing, I repeat difficult slides twice, okay. I encourage people who ask questions, but it also depends on how long that resource is. If there is too much content in that resource, then I might not answer your question straight away and might answer it at the end. And at the end of it, on every Wednesday you have a tutorial session also, a review session. I'll be there with sample questions and I'll go through these slides again, okay. And although my office hours have already been mentioned, you can walk into my office whenever you want. If I am available and I'm not busy, I will always take your questions, okay. I know right now all of you are preoccupied with the upcoming exams, but we cannot stop giving resources because the time is already very limited, okay. So kindly bear with me specifically, for the resources that I'll be giving before your exam. Again, my portion is not coming in the midterm, okay. Any questions you want to ask me? One thing more. Over the years, this has been a practice that there are students who just come in for attendance and they don't want to sit through the resource. Totally fine with me. I don't judge anybody on that. In fact, I am happy to have those students who really want to listen to what I have to say, okay. But whoever is leaving, just make sure you do it quietly without disturbing the dynamics of the class, and preferably do it at the very beginning, okay. I guess people who wanted to leave, they have already left, but kindly pass on this message, okay. So with that, let's start the first resource. Any questions, anybody? All right. So let's talk about today how proteins are transported in our cells. Now you know what is the organelle that synthesizes protein in our cells? Ribosomes. So ribosomes, it makes proteins. Now those proteins need to go either outside the cell or inside the cell or into the organelle. So how it is decided which protein will go where? So we will look into that under the topic. You said ribosomes, they make protein. Yes, correctly. Are there any types for ribosomes? Good. So there are free ribosomes that are present freely in the cytosol, and then there are ribosomes which are bound to the membrane of the endoplasmic reticulum. Now, the proteins that are made by free ribosomes and the proteins that are made by the attached ribosomes, do you think they go to the same destinations or there is some segregation in it? There is segregation, correct. So, any protein that is made by free ribosome, it will become part of the cytoplasm, it will become part of the nucleus, mitochondria and peroxisomes. So, generally speaking, proteins made by free ribosomes go to organelles, most of them. However, the proteins that are made by ribosomes that are bound to the ER, they often get associated with membranes. For example, membrane of ER, membrane of Golgi complex, and plasma membrane. Also, such proteins, they become part of lysosomes also. It is not mentioned here, in the later slides it is mentioned. Okay. So, in this slide it tells you that there is segregation. Now, let us look at transportation or trafficking of proteins that are synthesized by ribosomes, which get attached to the endoplasmic reticulum. Is the diagram visible to all of you? Because now I am going to only talk on the diagram. Okay. So, if you see in the diagram, there are 5 steps. Okay. So, what happens that, for example, a protein needs to be synthesized. Initially, all ribosomes are free. Okay. And what happens? An mRNA will come and bind to a free ribosome, and it will start making a protein. Now, as it is making a protein, until now the ribosome is free. Okay. As it is making a protein, the N terminal comes up. You have taken translation, you know when the protein is synthesized, it is the N terminal that comes up, correct? So, as the N terminal comes up, at the top of it, it has a sequence present which is called as signal sequence. So, as it is coming up, it has a tag on it. Okay. This signal sequence is recognized with a big protein that is present in the cytosol, and that protein is called as SRP, signal recognition particle. Now, this protein will bind to the signal sequence, as you see in step 2. It binds to the signal sequence, and since this protein is so big, it will drag the whole complex towards the endoplasmic reticulum membrane. So, SRP will drag the whole complex to bind to the receptor of SRP, which is present on the ER membrane. Now, the whole complex binds near the translocation complex. Translocation complex is a scientific term for an opening present in the ER membrane. So, ER membrane has these small openings which are called as translocation complexes. Okay. Now, once everything is bound to the membrane, the ribosome continues to make protein, as you can see it. However, the protein that is getting made, it starts entering inside the ER. Once the protein synthesis is finished, the ribosome gets removed and the protein completely enters inside the ER. The signal sequence also gets removed. Okay. And now that protein is free in the ER, it might undergo some modification in the ER. It can stay in the ER, or it can go further to other destinations. Okay. I repeat this again. So, what happens? Initially, there is a free ribosome binding to an mRNA. Okay. The ribosome starts translation and the protein starts popping up with the signal sequence on top of it. That signal sequence is recognized by a protein called as SRP, which is signal recognition particle. This protein binds to the signal sequence and it drags the whole complex near the translocation complex of the ER, binding to the SRP receptor. Okay. Now, everything is bound to the ER near an opening, which is translocation complex. The ribosome keeps on making the protein keeps on entering inside the ER. Once the translation is complete, ribosome it detaches, signal sequence gets detached, the protein is free inside the ER to be modified again, to be used by ER or to go to other destinations. Clear? Everything that I just said is mentioned in the text. Okay? Protein modification in the lumen of the ER. I just mentioned that some modification can take place for the protein. Now what happens if you look at the picture, focus on the picture again. This is what you see here is the ribosome, and this thread like structure that is coming in is actually the protein that is getting formed. So as it is entering the lumen of the endoplasmic reticulum, at the same time modification also starts happening for the protein. So since translation and modification is happening side by side, it is called as co-translational process. Now only yesterday you took modifications of the protein, correct? Which is the most common type of modification that happens to proteins? Glycosylation. Okay? So here also glycosylation will happen for this protein. So as the protein is entering inside, in the membrane there is a lipid molecule present which is called as dolicole. Again everything is mentioned in the text, so just focus here. This lipid molecule is present which is called as dolicole, and to this lipid molecule a whole chain of different sugars is attached. This is the chain of different sugars which is called as oligosaccharide. Oligo means many, saccharide means sugars. So what happens as the protein is entering, then dolicole will donate all its sugar to the protein that is getting synthesized, and the sugar gets attached to that protein. Okay? So this is the modification that happens. Now question, can this chain of sugar be attached to the protein randomly anywhere, or a specific sequence is needed? A specific sequence is needed. Always remember, in nature nothing happens randomly. There is always an order. Okay? So similarly, for the sugars to be attached to the new protein, there is a sequence needed. Now proteins are made up of what? They are made by ribosomes correctly, but they are made of amino acids. So this chain of sugars, it gets attached to a specific sequence of amino acid, and that sequence is either SN, SIR or SN, THR. Now the X that you see in the middle means any amino acid, but the amino acid on the left hand side is SN, and on the right hand side either it should be THR or it should be SIR. I am not giving you full forms of the amino acid right now, because in second semester you will take biochemistry, you will be given the full forms there. Okay? So this sugar, it gets attached to the SN portion of the sequence. As you see in the picture also, it is highlighting SN. Correct? Since it gets attached to the SN part of the sequence, this glycosylation is called as N-glycosylation. Okay? Also, glycosylation occurs in the Golgi apparatus as well, but that glycosylation is called as O-linked glycosylation. And what happens? In this glycosylation also, sugars are added, but they are not added to SN, rather they are added to the SIR portion of the sequence or the THR portion of the sequence. Okay? So when the sugar is added to SIR or THR, that is called as O-linked glycosylation, and that happens in the Golgi. And when the sugar is added to the SN part of it, it happens in the ER, and it is called as N-glycosylation. Clear to everybody? Yes? You have a question? No? Okay. Alright. So once the proteins have been modified in the ER, they will pass through the lumen of the ER. They can function inside the ER, and if they do not function in the ER, they need to be transported to another destination. Now if you go by sequence of the cell structure, can you enlighten me? Usually after the endoplasmic reticulum, what is the next organelle? Golgi apparatus. Very good. So from the ER, the next destination for a new protein is Golgi apparatus. What are the different portions of the Golgi? Yes. So there is cis and there is trans, and some of the portion in the middle is called medial. Again, not important, I am just telling you, okay, because I know you have been told only two, cis and trans. So what happens, a protein that needs to be transported to Golgi, it comes at the end of the ER, and the membrane of the ER forms a vesicle around it, okay. Now this vesicle will carry the protein to the Golgi, specifically cis Golgi, okay, and this vesicle will fuse with the cis and the protein gets released in the Golgi. Now question again, do you think the vesicle carrying the protein can randomly fuse anywhere on the cis, or there is a specific region for the fusion? There is a specific region, and how it is decided? Correct, markers. So if you focus on the lower picture, the donor organelle here is endoplasmic reticulum, okay. So as the vesicle is getting formed, it gets coated with a protein, and that coating is called as V-SNAR or vesicle SNAR. The red coating that you see here is the V-SNAR. Now once the V-SNAR coated vesicle is coming towards the acceptor organelle, the acceptor organelle will have a coating on it in a specific region, and that coating is called as T-SNAR, target SNAR. So wherever that target SNAR is present on cis Golgi, only there that vesicle will go and bind, okay. Always remember. So a vesicle coming off from the ER will have coating of V-SNAR, and the cis Golgi will have a coating of T-SNAR. Now let's talk about what happens to proteins in the Golgi complex. All of you know Golgi, that it has stacks. What are those stacks called? What is each of that stack called? Christy? So, you know, each of that stack is filled with enzymes, okay, and Golgi is known to sort everything and then send it to its destination. So something will enter through cis, it will go through some modification, it will come to the trans, and in the trans the sorting happens. So something that needs to go, for example, outside the cell will come in one region. Something, for example, needs to go to the plasma membrane, it will come in one region, and something needs to go to the lysosome, it comes in the other region. So sorting happens. So as I mentioned, the proteins, as they move through the Golgi stacks, each stack is filled with specific enzymes, and these enzymes, what they do with the protein is they add specific modification to those proteins depending on to which destination that protein is going to. For example, I am giving you an example of proteins that need to work in the lysosome. Such proteins need to have a phosphate tag on a mannose sugar. Phosphorylated means adding a phosphate on mannose. So any protein that has to be going to the lysosome gets a mannose in the ER, it comes to the Golgi gives it a phosphate on its mannose. So it becomes mannose 6 phosphate. So any protein which has mannose 6 phosphate, trans Golgi knows, oh, this needs to go to lysosome. This can't go to the cell membrane. Okay? So it's like an email address on that protein. And the other type of modification I have already explained, O-linked glycosylation also happens in Golgi. Again, glycosylation is addition of sugars. They get attached to the same sequence SNX-SER or SNX-THER, but instead of on SN it gets attached to SER or THER. That is why it is called as O-linked glycosylation. Correct? Now, as I mentioned, everything goes to trans Golgi where final sorting and packing happens. So some will stay back in the Golgi, other proteins will go outside. Again, everything is decided by the tag. What tag is there? Okay? And they go outside, they go to cell membrane, they go to lysosome. Clear to everybody? Okay. This is just an animation to show you that from the ER it comes in a vesicle to the Golgi, and within the Golgi stacks also it has to be a part of a vesicle to get transported from one stack to the other stack. And the same concept applies. As the vesicle is budding out, a V snar has to be there, and wherever that vesicle is fusing, a T snar has to be there. Okay. Okay, let's talk about lysosomes. Now, any protein that has to be part of the lysosome doesn't come from free ribosomes. It comes from ribosomes attached to the ER membrane. Can you enlighten me? What are lysosomes? Okay, very good. They are structures that degrade organelles. Yes? Very good. Yeah, so they are, anything else anybody wants to add? Yes? Yes. So basically if we look at lysosomes, they are like mincing machines, correct? So anything that is old, anything that needs to be broken down, it goes to the lysosome, and as he mentioned, the pH is acidic, and you mentioned that enzymes are there, they break those things down. So the enzymes that are involved in that breakage are called as acid hydrolases. See, I have given two different spellings, hydrolase here and a different hydrolase here. It's the same thing, don't get confused. Okay. So acid hydrolases are those class of enzymes. Acid means working in an acidic medium. Hydrolase means something, an enzyme that breaks something. Okay. You have any question? Okay. So let me say the story on the black picture given here. Okay. So what you see here is the protein coming from the RER. So let me go a little back. So a ribosome attaches to the rough endoplasmic reticulum. It makes the acid hydrolase protein. Okay. This acid hydrolase enzyme, it gets a mannose in the RER. Okay. Now this acid hydrolase from the RER, it enters into the Golgi. This whole stack that you see is Golgi. Okay. As it enters into the Golgi, a phosphate is added to it. Not anywhere on acid hydrolase, but on the mannose molecule that is attached to the enzyme. As a result, the mannose becomes mannose 6 phosphate. So what you see here is an acid hydrolase enzyme with a mannose-6-phosphate tag on it. Now this enzyme will slowly start moving towards the trans section of the Golgi where sorting happens. See, in cis and medial, the modification happens and the sorting happens in the trans Golgi. Now in the trans Golgi, in one of the section, if I take this as the trans Golgi, in one of these sections, there are receptors present which only and only recognizes mannose-6-phosphate. They do not know what is acid hydrolase, but they only recognize mannose-6-phosphate. So if a mannose-6-phosphate has a, if the acid hydrolase has a mannose-6-phosphate tag, it will bind here with the receptors as you see here. If any hydrolase has missed that tag, it will not get attached. Okay? Now when they are attached in one section, the same process, the membrane will bulge out, it will make a vesicle around those enzymes. See, even here the receptor name is mentioned, mannose-6, why is it showing like this? Mannose-6-phosphate receptor. Now as the vesicle gets formed, it forms with the help of a coating of a protein which is called as clathrin coat. You see blue dots? That is clathrin protein. Clathrin protein is that, the light is way too much to even make things visible here. So these blue dots that you see is the protein clathrin, which is important in making the vesicle. If everything is correct and clathrin is not there, the vesicle will not get formed or released. Now once the vesicle has been formed and released, the clathrin protein, it goes away, it is not needed. Okay? Now what happens, this vesicle carrying acid hydrolases attached to the receptor is slowly moving towards the cell membrane. At the same time, for example, some protein from outside of the cell has entered in the cell in an enclosed vesicle which is called as an endosome. It is moving from the right hand side. So from left hand side the enzymes are coming, from the right hand side the protein that needs to be degraded is coming, and the two vesicles, they fuse and they make a bigger vesicle here. Okay? This bigger vesicle is called as primary lysosome, and primary lysosome is inactive. Why? Because the enzymes are still attached to the receptor, they cannot function. Okay? I have not mentioned primary lysosome in the text. This is just to help you further understand. I am not going to ask you what is a primary or what is a secondary. Okay? Now what happens, to make the enzymes active and release them, the pH needs to be what? Acidic. So what happens, a pump, it starts working in this primary lysosome and starts pumping protons from the cytosol inside the lysosome. Do you remember what is that pump? I am sure you have been taught. V pump. V pump. Okay? It's the V pump that takes the protons or H plus ions from the cytosol and puts it into the lysosome. Now H plus ions will increase the acidity. As the environment becomes acidic, the enzymes, they get released from the receptor, the phosphate gets removed, the enzyme becomes active, it will start breaking whatever it needs to break. This is called as secondary lysosome. The receptors, they go back to the Golgi to do their job again. Okay? Let me explain it once more on a much bigger picture. But I need interaction coming from you also. Okay? So a protein that has been synthesized by a ribosome attached to ER, which is, and the protein would be acid hydrolase. It acquire what in the ER? It gets tagged with a sugar. What is that sugar? Mannose. So all acid hydrolase coming from the ER, they have a mannose tag. they enter into the Golgi. As they enter into the Golgi, what gets added to them? A phosphate. Where specifically? On the, yes, so mannose. So on the mannose the phosphate gets added, it becomes mannose 6 phosphate tag. So all the acid hydrolases which has this tag, they will go to the trans region and they will bind to the receptors for mannose 6 phosphate. So they get kind of blocked in one region of the trans Golgi. Now once they are blocked, the membrane starts forming a vesicle. However, something is needed for formation and release of that vesicle. What is that? A protein called as clathrin. So the blue dots that you see here is a clathrin coat. Now once the vesicle has been formed, this protein gets removed. Now vesicle containing the enzymes is moving towards the cell membrane. At the same time, some other protein enters into the cell, gets enclosed into a vesicle, and then both of them fuse. Now when they fuse, they make a bigger vesicle which is primary lysosome, although the enzymes are still not active. To make them active, a pump becomes functional, and that pump is V pump. And what does it pump? H plus ions from the cytosol into the lysosome. As the protons enter, the environment becomes acidic. As a result, the enzymes, they get released from the receptor. The phosphate also gets released. The enzymes become active. They start breaking whatever compound is there. The receptors, they go back to the trans Golgi to bring in, to make more lysosomes. Clear to everybody? Any questions? All right. Now, by now it is clear that any protein that is going out of the cell or to the cell membrane has to follow this route that we have just discussed in the last 35 minutes. However, the type of proteins that are secreted outside the cell is actually done by two important processes. And those two important processes are constitutive process and regulated process. Constitutive process is also called as structural secretion pathway. So don't get confused, okay? Constitutive and structural secretion is the same thing. And the other process is regulated secretion. Now the constitutive secretion happens for those proteins, and I am talking about, these are the secretions going outside the cell, okay. So constitutive happens for those proteins which are constantly needed by the outside environment of the cell, like the extracellular matrix, or in the blood the presence of albumin. So for constitutive secretion, the cell does not need any type of stimulus. This is something that happens automatically because the cell knows this particular protein is needed every hour or every day, or once in 2 weeks, okay. So the cell already knows that no stimulus is needed. However, on the other side, the regulated secretion happens only for those proteins which are needed for a particular process. If that process is happening, then those proteins will be secreted, and if that process is not happening, they will not get secreted. For example, this holds true for the digestive enzymes and the hormones. Now, all of you know about insulin. When is insulin secreted in our body? When the blood sugar is high, insulin gets secreted. This means the high blood sugar is a stimulus for pancreas to secrete insulin. Otherwise insulin, how would it be if insulin is getting secreted when we are in fasting state? It will be dangerous for us, correct? So insulin will get secreted only when blood sugar is high. Another example, we have salivary glands here and they have enzymes in the saliva. Normally when we are not eating something, those enzymes are not secreted in our mouth because they are so potent that if they are secreted otherwise, they can disturb the anatomy of our mouth. However, when you start chewing something, when you start eating something, the saliva starts getting secreted, which is filled with enzyme, and those enzymes will start working on that food. So that chewing that you are doing is a stimulus. So that activates the regulatory secretion pathway. Clear? Any questions? I would like to stop here. Okay. Tomorrow I meet you again. Also please note, whatever I am teaching you for now is not part of your midterm exam. Okay. Thank you for your attention.